Let's take a look at one of those hard OCR A-level biology topics, animal cloning, specifically somatic cell nuclear transfer. This is our process of animal cloning and how Dolly the sheep was produced. First, we're going to take a cell from the animal we want to clone. This cell contains a nucleus, and that nucleus has the full genome of that organism. But that cell may have differentiated, so we need to remove the nucleus from the cell. We also need another sheep, and from that sheep we are going to take an embryo, we're going to take an egg cell. We're going to remove the egg cell from that sheep, and that is going to produce our enucleated cell. Then we need to put the nucleus from the sheep we want to clone into the enucleated cell. And what we end up with is our cloned cell. This process is quite tricky and requires electrofusion, a small electric current to encourage the donor cell, the enucleated cell, to accept and take in that nucleus. This cloned cell is then going to be implanted into the surrogate sheep. Inside the surrogate sheep, that will develop into an embryo and into a fetus, and eventually the surrogate sheep will give birth to a little baby lamb. And this little baby lamb will be genetically identical to the original cloned parent. This is our process of animal cloning. We also have our process of embryo division and embryo cloning to produce identical offspring and identical twins. Follow for more advice and explanations of A-level biology.